We're really very lucky today to have such an excellent doctor, so well versed on this topic. We have Dr. Eli Katz. He is the Chief Medical Officer of eGenesis. It's a biotechnology company. Dr. Katz is an experienced transplant surgeon and a leader of clinical development in the pharmaceutical industry. He oversaw the development of therapeutics to modulate the immune system in autoimmune diseases and organ transplantations. Thank you so much, Dr. Katz, for being here today, and welcome. And I now turn the presentation over to you.
Thank you. Good morning, everybody. It's really, a nice way to spend Saturday morning to talk to patients who are experiencing their life organ transplantation and understand some of the issues around it. And I hope I can give you an overview of where we are these days with this exciting topic of xenotransplantation. As was mentioned, I myself, for quite many years, I was a liver transplant surgeon. I did liver transplantation as well as kidneys and pancreases in different centers in the US and in Israel. And the last about 12, 15 years, I was working in the industry, in the pharmaceutical industry, in development of different treatment for autoimmune diseases mentioned and transplantation. So I started my journey at eGenesis not long ago. It's about 2.5 months. But when I was approached with this potential position, I got very excited because xenotransplantation, as I wrote it here, is the future of organ transplantation, ? Or is it going to be the future of organ transplantation? In other words, I want to move from the question mark into the exclamation mark. Instead of asking the question, I hope we get to the point when we say, actually, yes, xenotransplantation is the future and is here to serve patients all over the world. So xenotransplantation is not something new. The concept of trying to use animal tissue or animal organs to try to address diseases and problems in humans is going back quite a bit, and it was done in different ways. Some of them quite, if you look at today, you probably will smile at that. For example, the Serge Vonorov in 1920 was trying to transplant testicles from non-human primate into humans to counterparts for rejuvenation. There was other trials, I think one of the famous ones is, of course, the 1984, the baby Faye that was born, a small infant is born with what we call left heart syndrome, when the left heart is not well developed, and if you don't do something, this baby usually die, even today. So there was a trial to give baboon heart to this baby, and it survived for 21 days, and it, of course, ended up with not becoming, at that time, the available treatment. There was the use of pig heart valve for many years successfully. Pig tissue, for example, pig skin is being used and developed to try to develop, to treat burns. So there is a lot of experience of trying to, or at least in thinking, to try to use animal for the benefit of human. And of course, jumping to just a few months ago, and you are in the area of University of Maryland, there was this significant achievement by transplanting porcine heart, pig heart into a human being that live for about 56 days and unfortunately succumb at the end. But this is a very important step going forward. So one of course, the first question that's coming in mind is why it's so tough? What was the issues that prevent us to transplant or to use tissues and organs from animals into humans? Now, you all are transplant recipients, and you understand transplantation from your perspective, and you know that anyone who gets organ transplant need to get immunosuppression. And the immunosuppression, even when we transplant from human to human, is critical to address the issue of rejection. Our body, our immune system is built in such a very sophisticated way that every time you try to put a foreign body or foreign protein or foreign tissue into it, the immune system doesn't like it and rejects it. And the rejection of the organ was from the beginning a challenge in transplantation, even between one human to another. And if you look at about 1950, when the first kidney transplant was done, it actually was done between identical twins. So identical twins have no issue with foreign because they are the same. They have the same kind of structure, right? The tissue are the same. So it was a very important step to show that you can do the transplant, and there was no issue with rejection. And actually, the recipient lived for many years after that. And Dr. Mary, who did it in Boston, got a Nobel Prize after a few years for this achievement. Since then, of course, we developed different immunosuppression, some of them right now very sophisticated one, that try to address the issue of rejection between two human beings. And now, as you know, and you're living with organs now, I'm sure few of you for many years, the immunosuppression is really successful. There's some issues with that, some side effects, but overall, we're able to transplant organs between one human to another with immunosuppression therapy and with very good results. What is the issue that all of us are aware of? That although we are very successful in organ transplantation, we don't have enough organs. And the shortage of organs is a painful issue that everybody involved in transplantation encountered and aware of. I remember, when I was practicing, I remember this desperate waiting for organ to come because patient is very sick and we need the organ and it doesn't come, and some patients die, and some patients, fortunately, we get the organ and they survive. There are so many patients now on dialysis, for example, around the country and around the world, thousands of patients on dialysis. Kidney transplantation can offer them, totally different type of life, free of dialysis and living longer and living better. We don't have enough. We are transplanting a very small fraction of the people who needs it. So we need more organs. And the question is, how to get more organs. And the attention, again, in the last 2 decades was turned into the possibility to use organ from another animal. Now you can imagine if it's tough and it's not easy to transplant from human to human because of the immune system tend to reject the organ, it's much tougher because of we are not the same, much far away from evolutionary. So we are far away from monkeys, which are our cousins, but we are far away from pigs or from sheep or from dogs, etc. So every time there was a trial to go from animal into human, the immediate rejection, what we call the hyperacute rejection, was destroying the organ almost immediately. So it took some time to try to figure out what can we do in order to overcome this barrier, this ability to overcome the immune system aggressive attack on an organ who come from another animal. So that's kind of the story of the last decades, or the modern approach to xenotransplantation, that eGenesis is one of the company working on it. And the basic idea of that, because of the development of understanding the human genome and understanding genetics and ability to modify and work with genes with very advanced technologies, the idea developed and now is put in practice that we can take a pig, for example, and apply different genetic technique and on the pig, and make the pig more similar in his genetic presentation to human. In other words, make the organ of the pigs more humans. And if you use this type of description, and by doing that, we lower the barrier, and the immune system of the humans are going to hopefully were not so aggressive to reject the animal organ. So this is the concept. The concept is to take an animal, and most of the work lately is done on pigs because they are matching in size, and it's easy to grow them, and we know how to deal with them. So pigs became the target, and the last 10, 20 years, there's a lot of effort by many investigators to work and see how we can modify the pig, that the organs of the pig will be more so-called human, and we can use them for transplantation. So the concept is going to be revolutionary, and it's going to address different issues related to organ transplantation. Of course, what we're going to do, we'll be able to, if we are successful and we're able to do that, we will be able to supply organs almost unlimited to anyone who needs it. It's going to create a much easier logistically way to deal with organ transplantation, because everything can be planned and be scheduled. And overall, it's going to really totally revolutionize the concept of transplantation. Not only we can transplant more people, but we can probably expand the indication. People who are now we deem not suitable for transplantation, hopefully we can do them in a different way. So there is a lot of hope, and we're talking about mainly right now the transplantation of the heart, transplantation of the kidney, islet cell from the pancreas to address diabetes. Lungs and liver are, of course, on the horizon, but they are much more complex, and will take some time to figure those out. Of course, other tissue like to treat the cataract and use other tissue, as I said, skin from animals, all this can be used and improve overall patient life. But the main focus, of course, is on transplantation and providing organ for transplantation. So here, just schematically, you see here the concept. We have the regular transplantation in black here, and you see that, of course, the supply is limited, and that's the problem. 20 patients per day are dying waiting for transplant in the US. The question of quality of the donor, not always we have a control of who is the donor, and because it's done in a kind of emergency basis, it's tough sometimes to assess the donors. If you're doing xenotransplant, we have a consistent quality. We know what we're doing. We know how we're raising the pigs, where we keep them, how we make them, keep them, clean or not carry any infection, etc., etc. So it's much more controlled environment. And we're turning emergency surgeries into something that's more scheduled and planned. And overall, we believe that, you know, as I said, the future will be that we'll have organs for everybody who needs it, organs that are high quality and done in a kind of elective way that always have a, you know, a better result overall to patient onto the system. Of course, what happened, and all of you are aware of it, what happened in the last about half a year was kind of very exciting and stimulating. So after all this research, that I'm going to go into some details soon about how we're doing that, but all this research are trying to turn the pig into a potential organ donor. There were 3 events that happened in the last half a year that actually tried for the first time to see how, if we do that into the humans, how it's going to work. The first 2 were done, one in the University of Alabama and one in the University of in New York, in NYU, was the connecting, kind of transplanting, but connecting kidney that were genetically engineered and from a pig, and connect them to a brain dead donor, somebody who actually is usually a donor for organ donors for humans, but for whatever reason it cannot be used, and the family agree and give consent. So in 2 events in Alabama and in New York, the kidney was connected to brain death donor, and for about 1 or 2 or 1.5 days, the kidney was working, and a lot of data was generated by that, and most of the kidneys didn't develop what is the fear in xenotransplantation, which is the hyperacute rejection. So both kidneys were not rejected on the spot. They were working for the amount of time that the experiment was going on. And this gives us the sense that the way we editing the pig and the way we prepare it for transplantation hopefully will work. Up to this point, all the studies were done by transplanting pig kidney into non-human primate, into monkeys. This is the first time that these kidneys from the pig were connected to the blood circulation of a human being. And then, of course, came the real significant achievement, which is taking this pig heart that was edited and was made more human, so-called, and using it in Mr. Bennett, who was a very sick patient who needs a heart transplant, and give a consent. His family gave a consent to use a pig heart, and the transplant was done successfully. The patient lived for 56 days after that, and then he died. The reason of the death is not very clear, and they're still trying to figure it out. It seems on its face that there was no clear evidence of rejection of the heart. There may be other issues. Maybe you heard about the finding of what we call porcine CMV, which is a virus that also infects humans. But at this point, there is no any evidence that this virus that was found in the heart from the pig caused any disease or any damage to the heart. So the virus that was found, the porcine CMV, probably didn't play a role in the demise of the patient. But all these events in the last half a year, of course, are very significant, and the interest and the sense that maybe we're coming close to actually we're able to use pig organs for humans become more of a reality and create a lot of energy in the field. And of course, there's a lot of work we need to do still. We are not over all the barriers, but that definitely gives hope that we one day will be there. So what actually we are doing? The work is being done, and it's kind of amazing to think about it. All the work of editing is being done on cells that are taken, just for you kind of to figure it out. we use cell taken from the ear of a pig. So we take cells from the ear of the pig, and using a very advanced technology of gene editing, it's called CRISPR, maybe you heard about it, and other technologies, our scientists are able to go into the cell itself, one cell, and do the editing, and I will explain what we're doing in a minute, do the editing required. After the editing is required, this cell is being fused with oval that was taken from a female pig, and a pregnancy is being induced. And out of this pregnancy, a pig come out, and the pig tissues now contain all the gene editing that we did. And I explain in a minute what we're doing. TKO is related to triple knockout. This is we're knocking out 3 molecules of sugar that are presenting on the surface of pig cells and are the most aggressive trigger of the human immune system. So if you don't take them out and you put organ from a pig to human, the immune system immediately, as I said before, destroy the organ. By taking out these 3 molecules of the sugars on the surface of the pig, what we call triple knockout, we eliminate this barrier. Then we inserting several human genes into the genome of the pig in order for the pig cells and the pig organ to look or behave more human. And I explain in a minute what it means. And the other thing we're doing, one of the, as you know, and I'm sure you're aware, one of the fear of xenotransplantation is the transmission of infection from the pig into the human. One of the known viruses that can cause a problem is the retrovirus that is embedded in the genome of the pig. You know, any virus is a piece of genetic material like RNA or DNA. So the retrovirus, in this case, the porcine retrovirus, is embedded in the DNA, the genome of the pig. So with our technology, we're able to take it out. We take all the genome of the virus out of the cell of the pig. So we're getting a pig who has the triple knockout, the insertion of human genes, and taking out this virus, the retrovirus, take it away. After we get this pig, now we can take again cells from this pig and use it to generate, either via pregnancy and breeding, or through cloning, we can produce many pigs that carry this gene modification. And these pigs now can be the donor when we need a kidney, or we need a liver, or we need a heart. We can take it from these pigs, and these organs now includes all the genetic engineering that was done. Our product for the kidney, as you see here, it's called eGenesis 2784. Doesn't matter the number, but that's the kind of the overall the way the process is being done. Any question you have, please write it in the chat room, and I will try to address it while we're going or at the end. This is another more kind of a little bit more detailed explanation how all this is working. So as I said, number one, we are doing in a level of a 1 cell, porcine cell, we do the genetic engineering we need to do in order to achieve what I just said, which is taking out the 3 sugars that are a problem and inserting the human genes and take out the virus. This genetically modified cell is now being let to develop, to divide, and creating many cells like that. we take the cells, we fuse it with oocyte, with a egg of a female pig after the nucleus of this is removed. And from that, we creating the pig that then eventually going to be the organ donor for our transplantation. All this work, of course, is done between our scientists and our operation, and we partnership with a very top transplant center around the country. We're working with, for example, with Mass General Hospital in Boston. We're working with Duke Center in Durham, North Carolina. We work in University of Wisconsin, and we're working with others. So this is, of course, effort that requires a lot of collaboration and a lot of working together in order to be successful. Very complex and very, demanding as far as the resources and the knowledge. Here is another schematic presentation for you to understand kind of more what is being done. So, as I said, all this is done by genetic engineering technique, mainly the CRISPR. And if you look at the first one, it's under the retroviral risk, is what I said before, how we go in and remove the genome of the virus out of the genetic material of the pig cell. So, so the virus is not anymore there. Then we're going in again to address the issue of a hyperacute rejection. We do what I said before, the triple gene knockout. We're going in and using again the CRISPR and genetic engineering technology, we remove the 3 glucose molecules on the surface that are on the surface of the cells. What we're removing is not the molecule, we're removing the genes that have the information to produce these molecules. But when we take out the genes, then they cannot produce the molecules, and these 3 sugar molecules are not presented on the cell of the pig anymore. So the risk of hyperacute rejection is being eliminated. And then we are doing what I said before, inserting human genes. And we're inserting specific human genes into the pig genome that going to express human molecules that are going to help in getting the organ be accepted and not rejected over time. And this gene related to coagulation issues, because we want to avoid any problem with blood coagulation that sometimes play a role in this type of transplantation. So we put in coagulation molecules that are human, so it's not going to create an issue with the pig when the pig organ being put in. We insert genes that have effect on the way that the immune system is responding to this graft. It's called the innate immunity genes. We put some genes that are more specific to the immune system that, again, kind of shut off the ability of the immune system to respond to the pig as a foreigner. Because there's a human gene on it, the immune system look at it and say, actually, I know these guys, they are humans, so we don't need to destroy them. And there's a system that involving all the inflammatory and rejection process in our immune system. It's called the complement system. And we put complement also that is human complement. And again, it's helped to subside and to suppress this system to be activated, because when it's activated, it's causing a lot of issues and can lead to rejection and other complications of the organ being transplanted. So at the end of the day, we're getting, at the end of this process, this cell will sit on the right here, and it's called engineering cell. And this cell includes the knockout of the 3 genes that have the information to produce the 3 glucose molecules, including all about 6 or 7 human genes that have the function I just described, and eliminated the retrovirus from the pig gene. And this cell is now the source for which we're going to put it into a oocyte or egg of a female pig and create an embryo, create a pregnancy, and the pregnancy will deliver the pig that includes now in all its tissue all this genetic modification. You see any question at this point in the chat room? I don't see the chat room here, so.
Dr. Katz, there is one question. Why are pig ears chosen as the cell source?
You know, any cell in the body can be the source. It just, easy to access. It fits the requirement as far as the cell to be edited. But in theory, any cell in the body can be edited. This is more for convenient, and the fibroblasts, the cells are being used are kind of more tolerating the editing more than other cells. Some other cells are more sensitive. When you start to kind of do stuff to them, they may die basically. These cells are pretty stable and also stable in the culture when you put them in a dish and they culture and they multiply. So this is the reason. But in theory, as far as the theoretical aspect here, any cell can be edited. Okay, let's continue. If most questions we'll answer. So what we're trying to do now in eGenesis and other companies work on this the same. The heart they went to Maryland is from another company to work on it. It's called United Therapeutics, Revivico. But our program now includes to develop pigs that are going to be sourced for kidneys. The HUCO, it's human compatible. So we call it human compatible kidneys, human compatible hearts, human compatible islet cells. We start to look into livers also, not so much for transplantation because the liver is a very complex organ, but maybe to develop the liver and use it as a mean to support patients who are in a severe acute liver failure that need support for 1 or 2 or 3 days until they get liver offer and they can be transplanted. So to do an extra body perfusion through this pig liver to try to support them. So we start to looking into that also. And I hope that it will materialize. So right now we are really concentrating on the kidneys, on the hearts, islet cell, and starting to look into the liver for this capacity, as I just mentioned. I don't want to go through all these numbers here. You probably have the slide. You can look at it later. But we all know what the problem is. It's a huge number of patients are sick, waiting for transplantation on the list. Very few getting it, and the solution, if we can provide organs that are safe and clean, is going to be very meaningful for patient life. So to do all that, of course, it's a major undertaking, and it takes a lot of effort, a lot of funding, a lot of everything. What you see on the left is our labs that right now is in Cambridge, Massachusetts. And this is where our scientists are doing all the gene editing that I just described. It's a very meticulous, very complex process, and we gather really, and we're fortunate to have top-level scientists that have experience with this technology and doing it, and we're now doing it on a regular basis. Of course, to be able to have pigs that are suitable for human use, we need to have facilities that take care of the pigs, raise them in a special way, that are free of any pathogens, that are getting the right treatment, that we need to have operating room there to be able to, what we're doing, we sometimes be using laparoscopic surgery to take the eggs from the female pig. We need operating room for that. We need operating room, all the deliveries done in cesarean section, so we need operating room for that. And eventually, when we start to supply organs, we will need each pig that's supposed to be a donor needs to be quarantined for about 2-3 weeks by itself before organs will take him away from him, in order to make sure that there is no any pathogen or any disease involved in that. So it takes a lot of resources to keep this pig production and pig growing and everything else in the right way. We have a large facility in Wisconsin, and we're building now what we call clean facility in Indiana. But this is ongoing effort, and the more we advance, the more we need this to be up to date, the state-of-the-art facilities. As I mentioned, we're working with different transplant centers. So you can see that this is a very complex and multifaceted kind of operation. So the kidney, that it's, as we mentioned, the kidney was the first organ that was done, so it's going probably to be, from our perspective, the first one that we probably going to use in humans. So as I said before, all our studies right now are done by transplanting a pig kidney into a monkey. And what we call a clinical payload, and the number is just the pig that has all the modification I just described, the one that we put in. And this is the number we give. We call it each genetic engineering, we call it a payload. And there's different ways to do, or different gene. For example, the human gene, you can insert different human genes. So each payload gets a number, but it doesn't matter. At the end of the day, we have editing organ that going to go into the monkey in our studies right now. And as you can see now, we have, and I'll show you in a minute the numbers of it, but you see that we are now have monkeys that survive between 177 days to 562 days after transplantation with a pig kidney. Now, the monkeys depend solely on the pig kidney, because in the operation, when we do the transplant, we take the kidneys of the monkey out, because we want to make sure that the pig kidney is the one that keeps the monkey alive and well. And if you see here, if you have now almost 2 years now of monkeys going with this kidney, it gives us really hope that we are doing something important here. And the question is now, from the FDA perspective and for our perspective, how many monkeys try like this we need to do, what we need to show, there are different aspects in order to say, okay, we're ready to go to patients. And when we get ready to go to patient, the other question, of course, will be, who are the first patients that may be suitable and willing to take part in this important experiment? Because the first few that we're going to do are going to be experiments. We need to try and see if everything we did and everything that seemed like work in monkeys going to work in humans. So we tried different scenarios of the gene editing and did different studies, but at the end of the day, everything is aimed into create data and basis to say, okay, we may be ready to go to humans. And in this regard, as you see in the last bullet point here, we actually submitted to the FDA a pretty thick booklet, explain everything we're doing and everything we did so far. And we're going to have a discussion with the FDA on July 22nd to see if we can bring the minds together and agree on if what was done is enough, should we do more, and what is the plan in order really to start the first trial in human beings. The difference between the kidney and, for example, the heart that was done in Maryland is, as you know, the heart was a life and death situation. If the patient doesn't get it, he is dead. And it was approved by the FDA under what we call a compassionate use. It's emergency use in emergency cases. It's different in kidneys. The kidney, we have dialysis. So patients are not in life and death situation because we support them with dialysis. So we cannot do the kidney under emergency kind of situation. We need to come to agreement with the FDA, and we need to have the data that we believe it's sufficient to bring this kidney transplant in a safe and good way to patients. We will learn a lot from the first study, and as I said, it's going to be experiment, but we need to do it in order to move forward. And if we're successful, of course, it will open the opportunities and make it more feasible to really move forward. So again, we coming back to the kidney and what, as I said, what we're doing with the non-human primate model, as I mentioned before, we remove the kidneys, as you see on the second plate here, and put one kidneys in. And the monkeys, many of them are living quite a long time. There's of course complication, not all of them survive. It's not an easy model to do it because monkeys, are not humans, and we don't not have all the facilities that we treat human with. But we get pretty good results up to at this point, and I think it's very encouraging. We're also looking at different immunosuppression and what immunosuppression may be important in xenotransplantation. All this information we're getting from these studies in the non-human primates. The next line shows you some of the things that we did. You see here, this is all these numbers here are monkeys that receive kidney from the pig. As I say, you see now there's a long ongoing monkeys that are surviving and feeling well. Some of them didn't. Some of them succumbed to different complications that we need to look at, understand, and learn from. Here we changed a little bit what we call the payload, as I explained to you, and we have now 4 that we did at this point. 2 are long, kind of going, and this one was also long, but then succumbed to some complication, and we need to keep going. So the question: how many of those transplant between the pig and the monkey you need to do in order to say, okay, we have good data, we understand what we need to do in order to go and do it in human beings. So that's the way you build your experience in transplant from a pig to monkey, and then this will serve as the basis to go to humans. We check on all of those, the creatinine, the BUN. If some of you, I'm sure, are kidney transplant recipient, you know what creatinine and BUN is. And as you see, most of these monkeys the function of the kidney is very good, to the point when they may succumb from other complications, and some of them do. But overall, we get a pretty good function. And as I said, some of them pretty long term after the transplant. So all this is encouraging data that we need to continue to collect, learn from that, draw the conclusion of what if we need to change anything, and move forward. A lot of work is being done. These studies are very, very complex. So in our mind, if everything goes well, you see here what our plans are. If everything goes well, and we are okay with donor production, with the immunosuppression regimen, we agree with the regulatory agencies on what needs to be done, and we're able to scale our operation up to produce what we need to produce, we believe that sometime at the end of 2023 or 2024, we probably will be able to start a human study of kidney transplantation. And of course, nothing is written in stone. This is kind of an assessment. If everything goes well according to the plan, there are going to be all kinds of issues sometime, and this may delay, maybe come short a little bit, but this is kind of the overall vision at this point of where we are as far as implementing this technology. With the heart, we'll follow kind of the same route. Or the genetic engineering is very similar, almost identical. The fact that the heart was done in University of Maryland, this heart, although come from different company, was almost identical to the heart that we are generating with our genetic engineering. So it's very similar. So it gives us a good kind of basis to be optimistic. We need to conduct quite a few. We did many in kidneys now, kidney to monkeys. We didn't know too many with heart. We need to do much more with heart to increase our experience to see the results. But other people in the last few years did quite a bit of heart transplantation in this model of pig heart into monkeys. Based on that, the physician at University of Maryland got the approval to do this emergency operation in Mr. Bennett. So we'll do all this work, the same similar, almost mimicking what we're doing in kidneys. And we hope to get also in the ability to do it in human beings. In the framework of probably from now, it will take about at least a year, probably 2 years until we are in a stage that we have enough data that we can say safely we can maybe go now into human beings. When we look at the hearts, we're also looking at the pediatric population. As I mentioned earlier, remember the baby who got baboon heart in 1981, Fay baby. There is a certain number of pediatric patients, very small babies, who born with a severe heart defects, where the left ventricle is not developed well, and they need immediate assistance, otherwise they're going to die. So in our plan to go forward with the heart, we're looking at two groups. One is the regular adults who need heart for transplantation, but we are going to look also into pediatrics, and we are now already in discussion with some cardiac surgeon who do pediatric transplantation to advance this and to get data by doing heart for small baby pigs into baby baboons and see if we can show that it's going to work, and then going to be the basis to do it in humans. So this is important work that we initiated lately and moving on that right now. The islet is another concept to use xenotransplantation. So diabetic mellitus is a major disease that unfortunately is common and causing in many times, if it's continue on a chronic level, it causes damage to the kidney to the point that many patients with diabetes end up needing kidney transplantation. So islet cell transplantation, islet cells are those cells in the pancreas that generating insulin. So in a normal human being, the pancreas has this, what we call islet cells, that when you eat nice ice cream or donuts with a lot of sugar, these islets are being triggered. They release insulin, and the insulin control the glucose in your blood and in your body. In patients with diabetes, these islets are not working well and not secreting the insulin they need to, and that's causing the diabetes and all the complications. So human human islet cells are being done for quite a while. So there is a process by which from donors who donate organs for transplantation, you take the pancreas and you produce the islet out of the pancreas and you infuse it into patients. And this is working quite well, but the process is very complex and you need few donors, few pancreases to able to generate enough cells, enough islet cells. So again, it's kind of shortage of islet cells. So because of that, the idea of using xeno islets is again compelling and maybe bring another solution to this issue. So we are using the same gene editing pigs to produce islet cells that carry all these gene editing qualities that I just mentioned. And you doing now, mainly at this point, we did a lot of studies in mice, but we are going to start doing some studies in monkeys. There is a way to create a model by which the monkey or the mice have so-called diabetes. They're having this issue with diabetes. They don't have insulin. And using cells, islet cells from our pigs in order to have the ability to infuse them to the patient. And hopefully they are going to produce insulin and take care of the issue of glucose metabolism and diabetes. Here you see some of the data from this study. Just look at the green one. This is the glucose high level before the islet was introduced, and then when open, it go down and maintain pretty low after the islets was injected. This is in a mice study. And here, when you look at the liver, because the cells are being put into the liver through a special cannula in the vein that goes into the liver, and you can see that there is, in this kind of staining, there are insulin-producing cells in the liver. So this cell that we put in kind of find home in the liver and sitting there and producing insulin. That's what we want to achieve. Again, it's not simple, and there's a lot of issues related to rejection and other issues of infusion into the liver, etc. But that's what hopefully we're going to do. As I said, we're doing now a lot in mice. Then we're going to move into models in monkeys. And if all this is going to work well, potentially you're going to have ability to treat patients with type 1 diabetes with infusion of this pig editing islet cells. So in general, a summary, we have a lot of effort now being done to move kidney forward, to move heart forward toward the islet cell, and there's a different meeting related to that with the FDA and others, different efforts in producing or building our facilities, our infrastructure, in order, as I said, that sometime, hopefully the end of 2023 or beginning of 2024, to start to see some clinical use of these organs from the pigs that we are producing. So this is some of our leadership team. Our CEO is Mike Curtis. He's a very experienced industry developer, a very experienced scientist. He's now leading our company, leading eGenesis. Jennifer is our strategic chief. Elizabeth is our HR that is in charge of getting the right talent that we need. Peter is our chief operating officer. And I don't need to introduce this guy. And as you see, we're working with different companies, different collaboration, different academic institutions. So it's really take a community. We need everybody to work together. We also try to develop working relationship and try to help each other between the different companies who are doing that, because some of the issues are general issues that we believe the company can work and should work together to find common solutions. And that's, I think, as far as what I want to present to you. I know I throw at you a lot of material and a lot of issues, but I think we have some time for questions, so don't be shy to ask questions.
That's great, Dr. Katz. It's fascinating. It's beyond my brain how you started this and how you continue with this. And if you could take down your screen, that would be great. We could see everybody's face, but we do have some questions, and we do have someone that has their hands up to ask you a question. Thank you. Let me go back to the chat. So if when these genetically modified organs become available to those awaiting transplants, what, if any, pre-transplant surgery, tests, and procedures will patient recipients need to do beyond what is done for non-genetically modified organs being transplanted?
The short question, the short answer is that probably not much in addition to what's done now. The only effort that we're going to implement that it's in addition to what is going now is monitoring carefully the recipient of the organ and probably his close family members to make sure, by taking basically blood samples and saliva samples, just to monitor and make sure there is no transfer of any pathogen, any kind of infection that we don't know about or we're not aware about. So the monitor of the patient after the xenotransplant is important to understand and to make sure we know and we can address any transfer of potential organ. We try to avoid it by doing everything I said we're doing. We're going to test the pig A to Z. We're going to keep him in a clean facility. We're going to test them before the transplant. We're going to keep them quarantined before the transplant. So there's a lot of effort done to keep the pig clean, but then we never know, right? So when it's going to the human, we're going to have a monitoring plan, quite long one, for almost for life, to make sure that we didn't induce any infection that we didn't know about. This is the main thing. As far as the operation, as far as the post-transplant immunosuppression, as far as the regular monitoring of biopsy from time to time and do all kind of other, it's going to be very similar to what we're doing now. So there is no any specific procedure that you need to do in addition to what you do now. It's just going to be much more focus on trying to make sure that we're monitoring the patient well and we're aware of any potential infection or any other issue that we may don't know about in this transfer from pig to humans.
Thank you, Dr. Katz. Next question. What are the two main factors that would determine moving from animals to human trials in kidney transplantation?
You know, in order to move to human, we need, as a company and as a responsible company and physician and the FDA, need to have a quite substantial, what we call preclinical data. We need to have the data come from our experiments with monkeys and other data that related to how we prepare the pig, how to maintain the pig, and then the result of the study we're doing. We need to establish the right immunosuppression, any modification that are specific for xenotransplant that we need to put. If we do all that, and we have the data that shows that we are successful, this is going to be the basis to go to humans. It's like any other, if you think in any other drug or any other device developed in using human, it starts in the lab, then it's go to animal studies, and then it go to humans. Not all the answer can be obtained when you're in the lab or you're doing animal studies. So always, when we go to humans, we don't have all the answers because you can't. And many times you cannot really translate what you see in animals into what's going to happen in humans. So we do the best we can. We accumulate as much data as everybody agree. We and the FDA and others agree, and the experts in the field agree that this is probably enough to move into humans, knowing that we may have surprises and that we may not have all the data, but it's enough to go in and try. And then we learn from that and move forward.
Thank you, Dr. Katz. A question about the combination of xeno and other organ procurement procedures. Can you combine some?
Can I ask the full question? Hi there.
Sure.
Yes, please.
Just to clarify, by the way, thank you for the talk. As a science nerd, I really appreciate this. I haven't seen anything in research, and I wanted to see what you think about combining xenotransplantation with a process like decellularized organ scaffolding and using them concurrently to help the process of organs.
Yeah, so there's of course a lot of research and work done in creating scaphoids and then populate those with cells. And the crossroad between xeno and those efforts probably has to do with the ability to use cells from, let's say, pig, just let's assume, to populate this scaphoid structure that usually are being made from some, other materials. So this is part of another approach to create artificial organs, so-called. Okay, so instead of taking full organs from a pig, we are going to create people using now the 3D printing, to create scaphoid that looks like, let's say, a kidney, but it's just the shell, right? The structure, and then you populate it with cells, and then you hope that this will mature into actually functional organ. There's a lot of obstacle on the way, but the cross between this xeno, in my mind at this point, is the ability to use cells, as I mentioned, for example, the islet cells that we use already, cells, to use the cell as the genetic modified pig as a source for the cell to populate these scaphoids organs.
Yes, I understand that, but how? I mean, part of the decellularized scaffold process is washing organs. Maybe it could be from a pig donor, but obviously washing it that thoroughly will never minimize the risk of leftover cells. So this is just my thought of using a pig, let's say kidney scaffolding with the xenotransplantation process, because all the cells cannot be washed out. And that's why using them in combination. I mean, the matrix doesn't have to be built through 3D printing. It can actually be from a different animal.
Yeah, but as you can understand, there's two concepts of trying to create organs, right?
Mm-hmm.
One concept says we're going to modify the pig and take its organ and use it as xenotransplantation. The other concept say we're going to use different technology to create organ. We're going to, whatever the methodology you're using to get the scaphoid, you get the scaphoid of the organ, and then you need to populate it with cells. You can populate it with human cells. You can populate it. Maybe some people think about taking cells from the potential recipient, from the patient, populate them in the lab, and then populate the scaffold with this human cells. So if you put back this organ to the patient, it's not going to be rejected at all because it's its own cells. So but this is two different concepts of creating organs. One is the xeno, in other words, taking organs from different species and putting in the human. The other is different methodologies to create artificial organs, so-called, and then use this to transplant into a patient. If you look at it this way, I think it becomes clear each technology or each concept, what will require in order to move forward.
Okay, thank you for your answer. Appreciate that. And again, great talk. I appreciate it.
No problem.
Thank
Thank
you,
you.
Doctor. Bernard thought the information was fascinating and very interesting. He hopes it helps many, many people. And then the last question, what is the expected immunosuppression for xenotransplantation when you compare it to regular transplantation?
This is a great question, and this is one of the questions that cause us headaches every day. Because we're trying to understand really what is going to be needed. You know, one of the risks of giving too much immunosuppression is that you get over immunosuppressed and then you start to develop all kind of infections. So always in transplantation, since we started transplantation, we always need to balance between how much immunosuppression you give in order to prevent rejection, and the risk, if you give too much, you're going to start to have infections. So in xeno, because of the immunological barrier is higher because we're moving organs between distant species, of course, the tendency is to try to give more because we expect more rejection. So we're trying really to look at that and try to see if really how much more we need. There is one drug that was developed about 20 years ago and then was stopped because of some issues, and now it's coming back, that it's addressing some specific pathway in the immune system that may be important in xenotransplantation. And we're testing it now in our studies. And this drug may be the one that usually is not used in regular transplantation. They're going to be used in xeno, in addition to what usually you see in transplantation. So we believe that we're going to start with, some of you probably heard the names, with, for example, thymoglobulin to start the first few days after transplantation, then give probably some tacrolimus, like probably some of you getting it, some steroids. So it's going to be similar to what we're giving usually in kidney transplantation, probably with the addition of one or two specific drugs that we believe are important for the xeno graft. And we need to find the right balance, again, between not give too much and not give too little. And this, by the way, one of the important things we're going to learn when we start doing it in humans.
Okay, Dr. Katz, thank you. And thank you very much, everyone, for all your questions and interest. And thank you for your time, Doctor. It was a great presentation, and I'm sure there's going to be more to hear about this in the future.
Thank you so much for listening. And it's definitely an exciting issue. And I think we'll hear more about it in the next year, 2 years, 3 years. And hopefully it will materialize to something that will help patients. That's the whole idea.
Yeah.
So thank you for your time, and have a great Saturday.
Thank you, Dr. Katz. Thank everyone. We thank you for attending today. We love seeing you all again. Please check out our website, www.triomaryland.org.